If you spit out a piece of gum today, you are not merely creating litter. You are leaving behind a small biological confession. Saliva, mouth bacteria, traces of lunch, and enough human cells to make a forensic scientist unusually interested in your snack. Now imagine someone does this nearly 10,000 years ago. The gum survives. Their DNA survives with it. And thousands of years later, strangers in laboratory suits reconstruct details about the person who chewed it. This actually happened in Scandinavia. The objects were small, dark lumps of chewed birch pitch. They preserved some of the oldest human DNA recovered from the region, along with evidence about Stone Age mouths, food, tools, and daily life. But there is an important correction to make immediately. This was not chewing gum in the modern sense. There was no peppermint flavor, foil wrapper, or optimistic claim about fresh breath. Birch pitch is a black-brown, tar-like material made by heating birch bark under controlled conditions. Stone Age people used it as an adhesive. It could help attach stone blades to handles, secure small inserts in composite tools, or repair objects. It was prehistoric glue, produced through a process that required practical knowledge and careful handling. The problem was that pitch hardened as it cooled. Chewing may have warmed and softened it until it became workable again. Think of someone preparing a lump of adhesive while making or repairing a hunting tool, except the workbench is their own mouth. There are other possibilities. People may sometimes have chewed it simply because chewing things is satisfying. Birch pitch may also have been used for dental or medicinal reasons. Some of its compounds have antiseptic properties, and later ethnographic evidence describes related uses. But we cannot ask the chewers what they intended, so the exact reason remains uncertain. It may have been glue preparation. It may have been medicine. It may occasionally have been the Stone Age equivalent of chewing a pen cap while thinking. The oldest Scandinavian pieces in this story came from Huseby Klev, a coastal hunter-fisher-gatherer site in western Sweden. The archaeological context dates to roughly 10,000 years ago, when people were establishing themselves in landscapes opened by the retreat of the great ice sheets. Excavations uncovered stoneworking debris, bone and antler material, plant and animal remains, and more than 100 pieces of pitch. Many had the dark, glossy shape of something that had been chewed. Some still carried recognizable tooth impressions. Studies of tooth-marked pieces suggested that several chewers were children or teenagers. One set of impressions was consistent with someone around 12 to 14 years old. That is a startlingly ordinary detail. Behind the grand phrase, Mesolithic hunter-gatherer, is possibly a teenager chewing the equipment. Researchers analyzed several pitch lumps and recovered genome-wide human DNA from 3 individuals. The genetic evidence included both male and female chewers and connected them with the hunter-gatherer populations living in Scandinavia at the time. This mattered because ancient DNA usually comes from human remains, especially teeth and dense bones. Those remains are not available at every site. They may decay, never be found, or be inappropriate to sample. Yet a chewed object can preserve direct biological evidence that a particular person handled it. The gum becomes a strange bridge between genetics and archaeology. A stone blade tells us what people made. The DNA in the pitch can tell us something about the people making it. So how does spit survive for almost 10,000 years? When someone chews pitch, saliva and cells from the lining of the mouth become mixed into the sticky material. The pitch then hardens, trapping fragments of DNA inside. Its water-repelling and relatively hostile chemical environment can slow some of the microbial and chemical processes that destroy genetic material. The site helped too. At Huseby Klev, a layer of clay appears to have sealed and protected the archaeological material. This was not indestructible DNA floating effortlessly through time. It was damaged, fragmented ancient DNA, preserved by an unusually favorable combination of material and environment. Scientists also have to prove that the DNA is genuinely old. Modern contamination is an obvious problem when the subject is an object people once put in their mouths and modern people later dug up, washed, labeled, carried, and studied. Ancient DNA laboratories use protective clothing, filtered rooms, surface cleaning, blank samples, and other controls. Researchers look for characteristic patterns of molecular damage and short fragment lengths. They also compare the genetic results with what is already known about ancient populations. The aim is to distinguish a Mesolithic chewer from an archaeologist who sneezed near the collection in 1993. The Swedish pitch eventually revealed more than human ancestry. A later metagenomic study examined 3 pieces dated to between about 9,890 and 9,540 years before present. The researchers found microbial communities matching the human mouth, including bacteria associated with tooth decay and gum disease. Their model suggested that the chewers may have experienced an unhealthy oral microbiome and possible periodontitis. Stone Age life had no refined sugar or soda machines, but it was not a dental paradise. Bacteria did not wait for agriculture to discover the human mouth. The pitch also contained genetic traces linked to hazelnut, red deer, red fox, apple, trout, mallard, and other organisms. Some may reflect food recently eaten or processed in the mouth. Others could represent materials handled by people or organisms present in the surrounding environment. This evidence must be treated carefully. Finding red fox DNA does not prove that the chewer had just ordered fox for lunch. Closely related species can be difficult to separate. Ancient fragments are short, and environmental DNA can travel in complicated ways. The safest conclusion is that chewed pitch can preserve clues about diet, activity, and surroundings. It does not provide a perfectly itemized receipt from a Stone Age restaurant. And this is where 2 famous chewing gum discoveries are often blended together. The roughly 10,000-year-old pitch pieces came from Huseby Klev in Sweden. They produced ancient human DNA from multiple individuals and later revealed oral microbes and possible dietary or environmental traces. The complete ancient human genome came from a different piece of birch pitch found at Sylvholm on the Danish island of Lola. That sample was younger, at about 5,700 years old. No human remains had been recovered from the Sylvholm site, but the pitch contained an exceptionally high proportion of human DNA. Researchers reconstructed a complete genome at low sequencing depth and determined that the chewer was female. She was genetically closer to Western European hunter-gatherers than to the early farming populations entering the region. Genetic markers suggested that she likely had dark skin, dark brown hair, and blue eyes, a combination already known from other European hunter-gatherers. Researchers nicknamed her Lola, although that was not her real name, which remains unavailable for fairly obvious reasons. The same piece preserved an oral microbiome, traces of Epstein-Barr virus, and DNA from hazelnut and mallard. Those last 2 may have come from a recent meal, though May is doing important work in that sentence. This was not a photograph, a diary, or a magical reconstruction of one entire life. Genetic traits are probabilities. Food traces provide a moment, not a lifetime diet. Oral microbes suggest health conditions, but they do not give us a complete medical chart. Even so, the intimacy is remarkable. Traditional archaeology often gives us durable things: weapons, buildings, pottery, monuments, and graves. Chewed pitch preserves something far less formal. It captures a person in the middle of an action so routine that they probably gave it no thought at all. That is why these objects matter beyond the novelty of ancient gum. They record the people who were not buried with treasure, did not command armies, and did not expect to be remembered. A discarded lump can reveal who was present, what population they belonged to, what lived in their mouth, and perhaps what they had recently eaten. The approach is still expanding. In 2025, researchers combined ancient DNA and chemical residue analysis on 30 Neolithic birch tar artifacts from 9 sites around the Alps. They compared chewed pieces with tar used on tools and repaired containers. The point is no longer simply that one lucky lump preserved DNA. These sticky materials can become a broader archive of prehistoric behavior. There is something deeply human about the whole discovery. We imagine the distant past through dramatic events, yet much of life has always consisted of maintenance. People sharpened tools, repaired broken equipment, dealt with aching gums, occupied restless children, ate whatever was available, and absent-mindedly chewed things. Nearly 10,000 years ago, someone finished with a lump of pitch and left it behind. To them, it was probably rubbish. To us, it is saliva, ancestry, technology, food, disease, and a set of tooth marks frozen in one small black object. The oldest message from a human mouth was not a speech. It was gum somebody forgot to throw away properly.